So here we are looking at the aspects of genetic engineering, and we're going to be looking at the generally what the main principles are, right, and what the process is for really creating these genetically modified organisms, right? And then we're going to be looking at CRISPR as a defense mechanism in bacteria, and with its Cas9 gene, and then we're going to be looking now at the implications of genetic engineering. Right? So that's the general order. So the principles of restriction enzymes, right? So let's look at restriction enzymes first. Right, so restriction sequence, recognition sequence, right? Restriction enzymes are proteins that recognize specific sequences of nucleotides in DNA called recognition sequences or recognition sites. These recognition sequences are usually palindromic, meaning they read the same forwards and backwards. So here we have Escherichia coli. This restriction enzyme would be ECO R1, right? And this is our palindromic sequence, is our recognition site, right? Here we have a Bacillus species, right? Oh, this is amylolic Q. Right? I always just call it amylo, right? Or amylolic Q, right? Because this is a long word. So Bacillus amylo, right, or Bacillus species. Bacillus species tend to have BAMH1 or BAMH2 or something. So usually a BAMH1 is what we're going to be looking at in the syllabus. And this is its recognition site. And then we have a Haemophilus influenzae using HIND3 or HIND3, right? Not HIND, but HIND3. And this is its recognition site, right? So the arrows, symbols show where the DNA is cut. All right, so recognition, this is their different recognition sites, and our restriction enzymes we know cuts. So it's like molecular scissors, it cuts at those sites. All right. So the topic is going to continue to get increasingly complex. So if there's a question, please let me know. Cutting now. Once a restriction enzyme recognizes its specific recognition sequence, it binds to the DNA at that site and cuts the DNA at specific points within or near the recognition site. So this cutting results in the creation of fragments of DNA, sticky ends or blunt ends, depending on the type of restriction enzyme. So you can have what we call sticky ends or blunt ends. So we know the type of ends. So the sticky ends now, some restriction enzymes cut DNA in a staggered manner, right, leaving overhanging sequences of single-stranded DNA at the ends. These overhangs are complementary to each other and can easily anneal, right, with each other, or with other DNA fragments. So they can easily attach to them. Blunt ends now, right, these are from enzymes that cut just straight down the middle in a straight manner, resulting in blunt ends with no overhangs. So here now, right, looking at the sticky ends, right? So the same restriction enzymes can cut DNA producing sticky ends, which are unpaired DNA sequences. So we look at sticky ends, and here we're looking at this. So here we can see the sticky ends for this part now, the 5' and 3' strands. Okay. So let's look at how BAMH1 actually cuts. So BAMH1 cuts in this manner, right? The staggered manner. This is to produce sticky ends. So these are what we know as sticky ends. All right. So when they have the overhang there. And H1 or He3 or He3, right? This is one of the enzymes that actually create blunt ends. It cuts straight down the middle. Those are the differences between sticky ends and blunt ends. Applications now. We know the restriction enzymes are widely used in molecular biology, techniques, such as restriction fragment length polymorphism, RFLP or analysis, DNA cloning, and gene editing, for example, CRISPR, Cas9, right? By cutting DNA at specific sites, researchers can manipulate and study DNA sequences more precisely. Okay? Now, the principles of ligase enzymes are molecular glue. Right? Let's have a look at this. So the general principles, right? So DNA repair. Ligase enzymes are responsible for catalyzing the formation of our phosphodiester bonds between adjacent nucleotides in DNA strands. So it helps to recreate those bonds. They join fragments. So in molecular biology, DNA ligase is used to join or paste DNA fragments together. This is particularly useful after DNA can be cut by restriction enzymes, and researchers can now insert a new DNA fragment into a plasmid, creating recombinant DNA, right, and actually having that effect there. All right, there, let me see, there seems to be an issue. It's lagging. Okay, let me see.